morning, dear students. Today we are going to see about ribonucleic acid. In the former class, we have finished about DNA. DNA, what about its composition, different types of DNAs, all those things we have finished in the former video. In this video, we will learn about ribonucleic acid. This is another type of nucleic acid. Here, the monomeric unit is not deoxyribonucleotides. Instead, ribonucleotides. That's the main difference regarding the monomeric units. In the case of DNA, we know that it is deoxyribonucleotides. Here it is ribonucleotides. Then which all together will constitute this ribonucleotides in the same way as that of deoxyribonucleotides. Here also it is composed of a nitrogen base, a pentose sugar, and a phosphate group. The difference is that here the pentose sugar present is not deoxyribose, instead ribose sugar. So that is the difference. Clear? Then let's see what are the main difference between RNA and DNA. The first one is the one that I just mentioned you before. That is, in RNA, the pentose sugar is ribose sugar instead of deoxyribose that's present in DNA. That's the first difference. Then second, which are the bases that we can see in DNA? Now, they are adenine, guanine, cytosine, and thymine. But in the case of RNA, instead of this thymine, uracil is present. That is the next difference. Then what is the difference between uracil and thymine? You look in this picture. This is uracil and this is thymine. As we look into its structure, we can see that in thymine, there is a methyl group in fifth carbon. But in uracil, that methyl group is absent. So that is the minor difference between the uracil and thymine. Anyway, in RNA, instead of that thymine, uracil base is present. That is the second difference. Clear? Then what is the third difference? We know that the DNA is a double stranded structure, but RNA is always single stranded. That is another difference. And also, even if it is single stranded, it usually see twisted, folded to form its secondary and tertiary structures. And the next difference is that this RNA is not stable as that of DNA. Stability wise, it is weak when compared with the DNA. Next difference is that for synthesizing a daughter RNA, RNA cannot depend another RNA. Clear? For synthesizing a new RNA, RNA needs the help of a DNA. From a DNA only, an RNA can form. So, always it needs a DNA for the synthesis of new RNA. But in the case of DNA, DNA can use DNA itself as its template. So that is the next difference. Okay. The next one is that if we take the DNA content of so many cells of a single organism, that content in all the cells will be same. But regarding the RNA, the content of the RNA that present in one cell will not be the same in the other cell. So it's not constant all time. That is another difference. And also we can locate RNA in cytoplasm and nucleus. But DNA we can locate only inside the nucleus. That is another difference. And DNA is only one type. One type means some different conformations are there, but I means genuinely it is only one type, single type. But RNA functional wise also, with respect to function and with respect to structure, RNA are of different types. Which are those different types of RNA that we are seeing in the next section? Types of RNA. In prokaryotes and eukaryotes, there exist three different types of RNA. Which are they? Ribosomal RNA, transfer RNA, and messenger RNA or mRNA. Okay. So first we can learn about ribosomal RNA. As the name indicates, this RNA, where we can see this RNA inside the ribosome. And what is the role of this ribosomal RNA? To translate the mRNA, to assist it, to help in the translation of mRNA to synthesize proteins. Okay, so which all cells which are engaged, busy with the protein synthesis, in those cells the amount of ribosomal RNA will be more. And I have told you that this ribosomal RNA or rRNA is located inside ribosomes. And the ribosome, based on the sedimentation properties, two types exist. Two types means one type exists in prokaryote and another type exists in eukaryote. Prokaryotic ribosome, its sedimentation coefficient. Sedimentation coefficient means while doing centrifugation, the rate at which it settles, that is the sedimentation. Swedberg unit is Swedberg unit. I hope that you all know all these things. Anyway, based on the sedimentation properties, ribosomes of the prokaryote is 70S type. And they possess a large subunit and a small subunit in the case of both prokaryotes and eukaryotes. Let's take the case of prokaryotes. Prokaryotes ribosome is 70S, I have told you, and the subunits are 50S and 30S. And inside the 50S subunit, there present two rRNA, and 34 ribosomal proteins. Okay. And what about the small subunit? In the small subunit, that is the 30th subunit, here present only one ribosomal RNA and 21 ribosomal proteins. That's about the prokaryotes ribosomes. Then what about eukaryotes ribosome? Sedimentation coefficient, based on the sedimentation coefficient, eukaryotic ribosome is called the 80S, and it also possesses a 60S large subunit and 40S small subunit. Inside the 60S subunit, we can locate 3 rRNA and 49 ribosomal proteins. Inside the 40th subunit, we can locate 1 rRNA and 33 ribosomal proteins. Okay, so that's about different types of ribosomes and its rRNA composition. And for joining these two subunits to do translation, one more thing, it requires the presence of the ion magnesium. If magnesium ion is absent, then that assemblance or organization will not take place and that will affect translation. Okay, that's important. So don't forget about this ion, its importance. It helps. It is the ion, it is the agent that helps in the association between the large and small subunit of ribosomes. Then one remarkable feature of this ribosomal RNA is that it possesses a high degree of secondary structure. Why it possesses a high degree of secondary structure? Due to intramolecular hydrogen bonding. Means we know that RNA is single stranded, and as it falls back and so many intramolecular within inside the same RNA itself, so many hydrogen bonds are forming, and that will give a double-stranded stem regions as well as single-stranded loops for the ribosomal RNA. I'll show you that image. So this is the ribosomal RNA. We can see a double-stranded stem region as well as single-stranded loops. So, and this feature means that having that double-stranded stem regions and single-stranded loops, that feature, it is necessary to do what? To associate between that rRNA and ribosomal proteins inside the ribosome for its assemblance. This secondary, high degree of secondary structure is very necessary. Okay. Next RNA that we are going to learn is transfer RNA, or otherwise tRNA. Then what is this transfer RNA or tRNA? You look to that board. Transfer means this RNA can transfer something. What it is transferring? It is transferring amino acids. So it acts as adapters. It transfers amino acid to where? To the mRNA template. But that complementarity in the base pairing between tRNA and mRNA is mainly based on the complementarity between anticodon of tRNA and codon of mRNA. Then what is anticodon? I will explain to you later in the coming slides. Anyway, for this moment, just simply telling you, suppose this is a tRNA, and in this tRNA, three bases will be there, and these three bases that we can, so many other bases are present in other regions, but specifically those bases that we can locate here in this end, it is called the anticodon. It is called the anticodon. And that complementarity in base pairing between tRNA and mRNA lies between the specificity or complementarity between anticodon and the codon. Here also three base codon present. So their specificity, their complementarity is the main thing that is the main important thing that plays the main role in pairing between the tRNA and mRNA. Okay. Then how we can name a transfer RNA? We can name the transfer RNA based on the type of the amino acid charged to it or loaded to it. Suppose this tRNA is charged with alanine amino acid. Then this tRNA will be known as alanyl tRNA. We can write that in this way. tRNA, alanyl tRNA. That means this tRNA is charged with alanine amino acid. That means this tRNA can carry alanine against the correct codon in the mRNA. Then what is this iso-accepting tRNA? Iso means same. So suppose this is one tRNA. This is another one. And this one is charged with alanine, and this one also charged with alanine. So these two tRNAs are isoaccepting. That means those tRNAs that are charged with the same amino acids are called isoaccepting tRNA. Then for load this amino acid to tRNA, it requires the help of some enzyme. What's the name of that enzyme? Amino acyl tRNA synthetase. Is that enzyme? This is the enzyme that correctly load the correct amino acid to the tRNA. When this amino acid is loaded to the tRNA, then it doesn't have any role to correctly get added against the codon in the mRNA. There, what plays the lead role? Those sequence in the anticodon. The anticodon and the codon in the mRNA, their complementarity is the main factor that determines the specificity of pairing. Okay. Next is about the Cloverleaf model of tRNA. Cloverleaf model of tRNA means it is the secondary structure of tRNA. We know that tRNA is a single stranded helical RNA, and as it folded back and how intermolecular hydrogen bonding, then it will give this Cloverleaf model. Clear? The tRNA is a single-stranded chain, and as it fold back and how intermolecular hydrogen bonding, then it will attain this Cloverleaf model. So it is a secondary structure of tRNA. Cloverleaf model is the secondary structure of tRNA. And how it is attained, that I already told you. And this secondary structure of tRNA in this cloverleaf model, it is the most stablest form of tRNA. And this stablest secondary structure can again fold back and give a tertiary structure. Okay, so here we are going to learn about the features of the cloverleaf model of tRNA. Is very important. Okay, so I'm just going to show you a picture. This is the cloverleaf model. In this picture, we can see that this single helical RNA is folding so many times and attaining this structure. So let's learn about its features. Uh, First point, it is a single-stranded chain. And how many ribonucleotides will present in that single stranded chain? Around 75 to 82 ribonucleotides are present in single tRNA. And in that tRNA, next point is that it possesses so many unusual bases, means not normal bases or not normal nucleotides. Why? Why they become unnormal, means abnormal or unusual? Because of some modifications that happened post-transcriptionally. After the transcription of that tRNA, it is having some modifications of the standard base like adenine, guanine, cytosine, and uracil. As a result of that modification, these unusual bases are forming in tRNA and they have some significance. And which are the names of some unusual bases? They are inosine, thymine, pseudouracil, methyl guanosine, dihydroxyuracil, then methyl inosine. So many such type of unusual bases are present in the tRNA. For remembering the names of some of these unusual bases, I have created a code and I have uploaded in Rani's Easy Memory and another channel of mine. So those who are interested can go and watch that. Next, it possesses one AM. It's called amino acid AM. I will show you that. This is the amino acid AM. I'm telling about that arm. It is a double helical stem-like arm. Okay, and what is the peculiarity? And that arm accommodates both 3' and 5' end of the tRNA. So it accommodates both 5' and 3' ends of tRNA. And in all tRNA at the 3' end, the base sequence will be CCA. It is constant. It is conserved. You can see that. That in 3' end, the sequence always will be CCA in the case of all tRNA. It has some significance because the adenosine nucleotide that we can see at the end, its OH group is attaching with that specific amino acid that to be transferred to the mRNA. Okay, so that's important. This the adenosine of this CCA to that adenosine, it's to its OH group, the amino acid that need to be transferred is attaching. So that is the importance. Okay, then 3 loops are present. 3 loops means its names are T-C loops, T-C loop, dihydroxyuracil loop, and anticodon loop. Let's see about all those loops. First, the T-C loop. What about this T-C loop? I will show you. It has some unique base sequence like ribothymine, pseudouracil, cytosine. And that T-C loop is formed of 5 base pairs and a terminal loop with the 7 base. So this T-C loop is formed with the help of 5 base pairs. 1, 2, 3, 4, 5 base pairs and a terminal loop with 7. 1, 2, 3, 4, 5, 6, 7 bases. So that's about here base pairs, 5 base pairs. And in the loop, there is no pairing. Instead, 7 bases we can see. So that's about T-C loop. And what is its significance? It has an attachment site for what? For the ribosome. We know that ribosome is doing the translation. Ribosome is the main intermediate to perform the translation. So for that, there is an attachment site in T-C loop for ribosome. Next loop is dihydroxyuracil loop. Dihydroxyuracil loop, that loop also has an enzyme recognition site. Which type of enzyme recognition site? We have seen that it's an enzyme that is transferring the specific amino acid to the tRNA. Amino acid tRNA transferase. Amino acid right. tRNA synthetase. Amino acid tRNA synthetase. So for that enzyme, this dihydroxyuracil loop possesses a recognition site. Okay, that's about the importance of that loop. That means for every loop, there is some significance. Then the third and very important is anticodon arm. Anticodon arm is opposite to the amino acid attachment arm. And this anticodon arm possesses a triplet of bases in its loop. And that triplet codon is called anticodon, and this anticodon is actually interacting with the codon of mRNA. So that's all about the cloverleaf model of tRNA. Once more, I will show you the image of that cloverleaf model. So we have learned that an amino acid acceptor arm, because this arm itself is attaching with the amino acid, and that amino acid is specifically attaching to the OH of the terminal adenosine of CCA base sequence. And this is the T-C loop, and T-C loop, we have seen that that loop is formed of 5 base pairs and 7 bases, and it has a ribosome recognition site, and this is the anticodon loop, and this is the dihydroxyuracil loop. It possesses a binding or recognition site for the activating enzyme, or the enzyme that is transferring specific amino acid to tRNA. That's all about the cloverleaf model of tRNA. I hope that's very clear to you. Next, we are going to learn about messenger RNA. Messenger RNA means what? It is an intermediate molecule where we can see what is the role. It is involved in the conversion of the information stored or coded in the DNA to the amino acid sequence in the protein or polypeptide chain. So in that process, in between the pathway, we can locate the presence of this mRNA. So it is an intermediate molecule. And in this mRNA, the genetic message is carried in the form of a triplet of nucleotides like A, U, G. And this triplet correspond to the correct specific amino acid of the polypeptide chain. Okay, clear? So, and also this mRNA is so transient. Its lifespan is very short when compared to that of other RNAs like rRNA and tRNA. Next, we are going to see the prokaryotic mRNA or messenger RNA. Which are the parts of a prokaryotic mRNA? It is composed of 3 parts. First one, I am going to explain you about the middle coding sequence. They present a middle coding sequence, and this coding sequence is flanked by 2 untranslated region. It can be represented as UTR. 2 untranslated region in 2 ends. So, generally, that prokaryotic mRNA is composed of 3 parts. A middle coding region is flanked by 2 untranslated regions at both terminus. And the length of this coding region, it varies, that depends upon the length of the protein to be coded. So based on its length, the length of the coding region will vary. Then there's some specific terms for this UTR at both ends. At the 5' end, this UTR, untranslated region, is called the leader sequence. And what is the significance of this leader sequence? This leader sequence helps to recognize the ribosome for what? To initiate the protein synthesis. And also this 5' end has a triphosphate terminus. So that's the peculiarity, and that's the name of that. Then what is the name of that 3' end? 3' end is called the trailer sequence. The trailer sequence at the 3' terminus. So next I'm going to show you an image of prokaryotic mRNA. This is the prokaryotic mRNA. In this picture, you can see that middle coding region, it is flanked by the leader sequence and trailer sequence. Okay. And Next, we are going to learn about the eukaryotic mRNA. As we have just seen about the prokaryotic mRNA, now we can think about what are the difference when compared to that of prokaryotic mRNA. This eukaryotic mRNA have. What are the difference? What are the peculiarities? So in the case of eukaryotic mRNA, it is monocistronic. Monocistronic means something special regarding the prokaryotic mRNA also. There that mRNA is polycistronic. Cistronic. Then what do you mean by this monocistronic and polycistronic? First, let's understand what is monocistronic. Monocistronic mRNA means in that mRNA, it contains a continuous sequence of nucleotides and it codes for a single polypeptide chain. Then what is polycistronic? Polycistronic mRNA means it contains a continuous nucleotide sequence that is capable to code for multiple polypeptide chains. So that is polycistronic. So in the case of eukaryote, the mRNA is monocistronic. And this is the picture of eukaryotic mRNA. As we look to its 5' end, here along with that 5' UTR, we can see an additional structure, a cap at the 5' end. What is the name of that cap? Its name is 7-methyl guanosine cap. A cap is there for the protection of the 5' terminal end. That is another peculiarity. And as we look to the 3' end, there also along with the 3' UTR sequence, we can see an additional tail, a poly-A tail, polyadenylyl tail. That means at the 3' end, a multiple repeats of adenosine nucleotide we can see at the 3' end. So that is the another peculiarity when compared to that of prokaryotic mRNA. The untranslated regions are also present at 5' and 3' end that I have already shown to you. Then here, as this eukaryotic mRNA, as it get transcribed from the eukaryotic gene, that first formed mRNA is called, it has a specific name, it is called primary mRNA transcript. Why such a specific name for that first formed mRNA in the case of eukaryote? Because it contains some wanted and unwanted regions. That wanted regions are called exons and unwanted regions are called introns. Okay, there is some significance for these means introns also that you will learn in some other videos that you can locate in my channel itself. Anyway, wanted and unwanted regions are present. Because of that, it requires some processing before get transported outside the nucleus to the cytoplasm. Okay, so it requires some post-transcriptional processing. And after that post-transcriptional processing, then that means mature mRNA is transported to the cytoplasm for translation. That's all about eukaryotic mRNA. Picture we have seen here. Next, I'm going to show you the difference between eukaryotic mRNA and prokaryotic mRNA in the form of a table, because that table will help you if such a question came for your university examination. It contains some additional points too. So by that way, that area becomes more clear to you. So that's what I'm going to show you. So this is that table. In the case of this is regarding prokaryotic mRNA, this is eukaryotic mRNA. In the case of prokaryotic mRNA, that mRNA is produced from some operons. Then what is operons? Operons means they are a cluster of genes, and those cluster of genes are under the control of a single promoter. In the case of eukaryotes, they present only a single gene for one promoter. But in the case of this is the case of eukaryotes, sorry. And in the case of prokaryotes, a group of genes are there, and that cluster of genes will be under the control of a single promoter. Such type of that functional units are called operon. Is it clear? So this is the prokaryotic mRNA, this is eukaryotic mRNA. As it is formed in the case of prokaryotic mRNA, as it is formed from operon, that is why that single mRNA can code for multiple polypeptides. That means it is polycystonic. Here, as it is formed from a single gene, it is all capable to form only a single polypeptide gene, monocystonic. Okay, that point is clear. And next, coming back, in the case of eukaryote, that mRNA is produced from a single gene. Next difference, the synthesis of mRNA occurs in cytoplasm because in prokaryote there is no nucleus. So it's complete formation happens in cytoplasm itself, no other way. In the case of eukaryote, the mRNA is formed inside the nucleus, and after the formation and mature, it is transporting outside from the nucleus to the cytoplasm. In the case of prokaryote, it is polycystonic. Here it is monocystonic. And in the case of prokaryotes, that 5' cap and 3' tail both are absent, but in eukaryote both are present. In the case of prokaryote, as it is present in cytoplasm itself, means it is get transcribed in the cytoplasm itself, along with it for transcription, translation also going on. So it is coupled there. But in the case of eukaryotes, the transcription is happening inside the nucleus, and after that only it's possible to transport outside the nucleus to cytoplasm for the translation. So here that two process, transcription and translation, is not coupled. And here for recruiting the ribosome, in the case of prokaryote, that mRNA possesses a specific sequence called a Shine-Dalgarno sequence. But in the case of eukaryote, that 5' cap is recruiting that ribosome for initiating the translation. And lifespan with respect to that mRNA in the case of prokaryote, it's very short when compared to that of eukaryotic mRNA. And in case of prokaryote, that mRNA requires less post-transcriptional modifications. But in the case of eukaryote, as it processes so many unwanted regions like introns, it need more post-transcriptional modifications. So these are the differences, I think. It's very clear to you regarding almost all different types of RNAs in this section, like rRNA, tRNA, and mRNA. Then we can wind up here. Thank you.
I'm an icon, man. Ain't my girl about a 10. I should fuck her in the Benz.